Today's video is on cell respiration, and we're looking at the structure of the mitochondria. It is very important to understand the structure of this essential organelle within cellular respiration. It's involved within aerobic respiration, and its structure is vital to being an efficient way of eukaryotic cells processing this reaction. The reactions that occur within our mitochondria are the Krebs cycle and electron transport chain, which are parts which are essential in aerobic respiration. We need to understand that this structure is vital for all eukaryotic cells, whether that's plant cells or animal cells. So when we make pyruvate via glycolysis, it has two routes: the anaerobic, where it gets converted to lactic acid, and the aerobic, where it passes into mitochondria and can be further processed, creating more ATP. The amount of ATP that we produce in anaerobic is just 2, and the ATP within aerobic is 36. So it's vital to understand how aerobic respiration works. So here is a quick diagram of the overall structure of our mitochondria. We can see it has a number of key points that are being illustrated in our diagram. So there are 2 membranes, an outer and an inner membrane. It also has DNA and also ribosomes. So our outer membrane, very important, contains everything inside. Also, the space between the outer and the inner membrane, the intermembrane space, is vital for the electron transport chain part of respiration. The inner membrane, as we can see, is folded to form finger-like structures to increase the surface area so that more of the electron transport chain can occur. And these finger-like projections are referred to as cristae. So the cristae increase the surface area, allowing more aerobic respiration to occur. The cytoplasm within the mitochondria is known as the matrix, and it has its own DNA, as I mentioned. And this DNA is very much like prokaryotic DNA. It's circular and single loop, and it has no histones. Then the ribosomes that are found in the matrix are 70S ribosomes. And these are used to make all the enzymes and parts that are required for aerobic respiration. This strange structure brings up an idea of a very important theory, and that is Endosymbiotic Theory. So the Endosymbiotic Theory is the theory that originally mitochondria and chloroplasts were once free living prokaryotic cells. And then in the past, our ancient ancestors of eukaryotic cells engulfed the mitochondria and the chloroplasts. And instead of destroying it and using those nutrients, they entered a symbiotic relationship with these free living bacteria and basically evolved over time to create the mitochondria and the chloroplasts we know today. And the evidence is very much in the structure of our mitochondria. It has a double membrane, and this is unusual for organelles inside a eukaryote. So this would suggest that it was previously engulfed by endocytosis in its past. That's why it has an outer membrane. It has its own DNA, which is similar to prokaryotic DNA. So it controls what enzymes and what things are inside it. It has 70 S ribosomes. Why would that possibly have a different size ribosomes? Eukaryotic cells have 80 S ribosomes. There's no reason why we would just make 70 S ribosomes for our mitochondria. And they control their own replication, which is similar to binary fission. All this evidence supports the endosymbiotic theory. So, to review today's lesson, we learned the structure of a mitochondrion from the cristae and the matrix, the outer and inner membrane, its DNA and its ribosomes. And all these parts will play a role in aerobic respiration. And of course, the theory we just went over, the endosymbiotic theory, which is one of my favorite theories. And it's really quite fascinating at how this organelle evolved over time. So, any questions, any comments?